Hey.
Hello, everybody. Can everybody hear me? Very good. Very good. My name is Payami. I am a researcher at UAB. I'm going to minimize this thing and put it away so you can see my slides.
Alice, we figured this out. Okay.
Alice, you're trying to figure it out. I can hear you now. Can you hear me, Alice? You're going to be muted soon.
Yeah. There we go.
Okay.
All right.
So for some reason, I lost myself.
You there?
Oh, okay.
I see you.
You see me. Good.
Let's see if I have one that's
All right. So let me introduce myself. Off the bat, I should let you know that I'm not a physician. I do not have an MD. I'm a scientist with a PhD. I got my PhD from UC Berkeley in genetics. And then I have been doing research on Parkinson's and Alzheimer's for nearly 30 years. Oops. I lead a consortium. We call it Neurogenetics Research Consortium. Don't even bother Googling it because we're not into self-promotion. The consortium is actually me and many other scientists at movement disorder clinics in University of Washington, Seattle, Oregon Health Sciences University, Albany Medical College in New York, Emory University in Atlanta, and the University of Alabama at Birmingham, where I am right now. We have in our studies over 5,000 research volunteers, including persons with Parkinson's disease and people who do not have Parkinson's disease. I'm a professor of neurology and genetics at University of Alabama at Birmingham currently. And the research has been funded by grants from the National Institute of Health, Michael J. Fox Foundation for Parkinson's Disease, and US Army Department of Defense. So Parkinson's has usually been defined as a brain disease that causes movement disorder. The area that degenerates in the brain is the substantia nigra. And cells in the substantia nigra make dopamine. So when so many of the cells in substantia nigra die that the levels of dopamine drop to 20% or less, that's when the motor signs show up. That's the traditional view of Parkinson's, and it's accurate, but it's far from complete. Brian, I think you are the one who is showing up. Is that correct, or is it me? Does the audience see me or Brian?
We see both of you.
Both of us, okay,
Yes.
Cool. Because I could see only Brian. Thank you, Michael.
Sure.
So
I can see all three of you, Michael.
yeah, now it's 3, yeah.
No,
Brian, if you could limit that to 1 so it doesn't cover the slides, that would be great.
It's not covering the slides. It's just the way it Zoom sort of moves around based on sound and sight. But no, the slides, everybody can see the slides.
Okay.
I can't see that.
Okay.
I can't see the right side of the slides.
Yeah, it covers the slides.
Yeah, I see 4 people.
Okay.
So one thing you
Let's see
can
what we
do
got.
is
Hang on, let's do this.
If you go under,
how's that? How's
click-
that?
you can go under view and adjust where the slides and the people are, and that'll help.
right.
All right. How's
There's
that?
2 or 3 options, but you can.
Okay.
That's good.
That now shows Michael only. You want to switch it so it shows me? I'm kind of animated.
It's showing you.
It shows you, and the more you talk, the more you'll stay there.
Oh, okay, very good.
It's,
All right.
it's not what you're seeing of you isn't necessarily reflective of
Right, okay.
what's being displayed.
So this is very different from the talks that I've given on Zoom. So I just wanted to make sure that everybody's on board and everybody can see. Please speak up if you can. So the contemporary view on Parkinson's is that Parkinson's is not just one disease. And it's not just a brain disease, and it's not just a movement disorder. It's actually systemic. Well, we don't know what actually triggers the start of PD. The current thinking is that some triggers may target the brain first, and then they spread down to the body, and some triggers may target the gut first, and then spread up to the brain. So this slide is supposed to show that Parkinson's disease does not happen overnight. The neurodegeneration happens decades before you see the motor signs that ends up in diagnosis. Many decades before neurodegeneration starts by a trigger. There are probably more than one trigger. And then it's a silent period, and then non-motor symptoms start showing up, and then slight motor signs, and finally you get the cardinal signs of PD that ends up in the diagnosis. If you look at the same thing, but from the clinical symptom perspective, time 0 is the time of diagnosis. If you go backward, you see that there is loss of smell. Before that is REM sleep behavior disorder. Actually, people who have RBD have a very high likelihood of developing neurodegenerative disease, including Parkinson's disease. You can even go further back 20 years before the diagnosis, and you see a high incidence of constipation in people who end up with Parkinson's disease, but not even close to every person who's constipated develops Parkinson's, as opposed to RBD, that just about all of them develop Parkinson's. So we really don't know when Parkinson's actually starts. That actually goes to one of the questions that was posed to me before the talk. Can we prevent it? Well, we can't really prevent it because we don't know when it starts, but we could interfere at different levels. So one of the areas of research is now is to take cohorts of people who have RBD and see if we can interfere with RBD going into Parkinson's disease. I would like to tell you about Dr. Brock. He's a German scientist, very well known in the Parkinson's field, also Alzheimer's. He studied, he published 2 papers in 2003. In the first one, he actually looked at the spread of the pathology. So let me tell you what the pathology was. Alpha-synuclein is a protein that gets corrupted, and that corrupted alpha-synuclein is thought to be the beginnings of Parkinson's pathology. It's actually a hallmark of PD brain and used for diagnosis. So when Dr. Brock looked at the brain, he actually saw that there is pathology at the brain stem, and it goes up to the brain, not coming down. And in the second paper, he actually proposed his famous Brock hypothesis. He said that the disorder might originate outside the central nervous system, which is the brain, caused by an as yet unidentified pathogen that is capable of passing the mucosal barrier of gastrointestinal tract, that's lining of the gut, and via enteric neurons like the vagus nerve, came into the central nervous system and go to the brain. So basically he's saying that he thought that a pathogen, an infectious agent, gets into the gut, and then from the gut, it gets into the vagus nerve, which connects the gut to the brain, and that's how it gets to the brain and causes Parkinson's. Well, there has been a lot of support, a lot of literature in different aspects of Brock's hypothesis. Sure enough, pathology is seen in the gut before in the brain in many people. The lining of the gut is compromised. So many aspects of Brock's hypothesis have support in the literature, except that as yet unidentified pathogen was still at large and may still be, but our most recent paper shows that we have found some pathogens in the gut of Parkinson's. We're not sure that they're the cause, but it goes along with Brock's hypothesis, and I'll get to that. So for a long time, we have known that there's a connection between the gut and Parkinson's disease. So one of them is that GI problems, including constipation, inflammation in the gut, and what we call leaky gut, the membrane that encloses the gut and keeps the pathogens out and keeps everything else, keeps the gut intact. It's leaky. Things can come and go. So we have known that these problems actually precede the motor signs of PD. And more recently, it's been shown that in many people with Parkinson's, alpha-synuclein deposits, the hallmarks of PD that are used for diagnosis, actually may appear in the gut before they appear in the brain. And this was really fun when it came out. Alpha-synuclein can move around. It can spread from cell to cell, and it can spread from the gut into the nervous system and go up to the brain. So it's kind of adding to the evidence that PD may start in the gut and then spread to the brain. This doesn't mean that all PD starts in the gut and goes to the brain. The gut is actually called your second brain. The gut has a very complex system of neurons. It's called the enteric nervous system that connects directly to the brain via the vagus nerve. So, if some cases of Parkinson's start in the gut and spread to the brain via the vagus nerve, then one would think that people who have had their vagus nerve cut, they would have a lower risk of getting PD, right? And why would you cut the vagus nerve? Well, decades ago, people thought that that would cure ulcers, which was totally nonsense. But a lot of people got vagotomy because of that reason, and countries that have large national registries could go back and look at the people who had vagotomy and see if they ended up getting Parkinson's or not. So the first paper that came out was from Denmark, and the second paper was from Sweden. And the results were similar. The one in Denmark went back to 1977 to 1995. There were over 5,000 people who had vagotomies, means that their vagus nerve were completely severed. And then they looked 20 years later to see how many of those people developed Parkinson's as opposed to people who did not have vagotomy. It turned out that the rate of Parkinson's was half in people who had vagotomy as compared to people who didn't have vagotomy. Similarly, in Sweden, but they didn't have 20 years. They could go back to 1990 to 2010, but they showed that even after 5 years post-vagotomy, the risk of PD was reduced to half. So this kind of supports that something is going from the gut to the brain, and if you cut what's connecting the gut to the brain, you can reduce the risk, but you are not eliminating the risk altogether, which goes along with some cases maybe starting in the gut, going to the brain, but not all. This paper just came out. Brain first versus body first. We have been talking in the field about Parkinson's not being one disease, talking about Brock's hypothesis. Does it start in the gut? Does it start in the brain? This paper was actually very nice. It was in 2020, this year it came out. What they did was that they took persons who were newly diagnosed with Parkinson's disease, and then they imaged them, imaged their organs, the gut, the heart, the vagus nerve, and the brain. They imaged them for pathological markers of Parkinson's disease to see in each person where the pathology appears first and it's most severe, and they trace the severe and progression across the body. You realize, guys, that I'm actually going to show you data, so bear with me. So this is right out of the paper. So on the left, panel A, they saw that some of the patients showed the pathology first in the gut, and it's most severe in the gut. And with time, it spreads to the brain, to the heart, and then goes up to the brain, ends up in substantia nigra. But then in some other patients, they saw first the pathology in substantia nigra, and most severe there, and with time, it spread down to the gut. I'm going to pause a minute and make sure that I've got everybody with me. There are no major questions, and we can move from there.
Okay.
Is everybody with me?
Mhm.
Any questions? You can raise your hand, or
You also type questions in the chat box if you have them.
are we good? Am I clear? Is there anything I need to, I'm clear? Okay, I'm going to move on then.
All right.
So, if some forms of Parkinson's start in the gut, how does it start? And what starts it? What's in the gut? That brings me to microbiome. Microbiome refers to the microorganisms that live in our body. They are, most of them, they are harmless. They're in a symbiotic relationship with us. And without them, we'll probably be dead. They're essential for our health. There's a microbiome that's specific to the nose, to the skin, intestine, vagina, umbilical cord, mouth, armpit, breast milk, and feces here is actually what we use to get an estimation of what is in the gut. What does microbiome do? These microorganisms, of course, they help with digestion and metabolism, but they have even more important roles. They regulate the entire body's immune system and keep us, to some degree, safe from infections. They also help us metabolize drugs and toxins that we're exposed to. It's important for intestinal function, bone, and for our talk, most relevant is the connection between the microbiome in the gut and the brain and the behavior. So, if one is going to study microbiome, I think I want to set the stage by showing you how mind-boggling It is both in sheer numbers and the diversity. In my gut, there are 10 trillion microorganisms. Compare that to 7.8 billion people living in the world. 1 trillion is 1,000 billion. So imagine how many bugs there are in the gut. Not only it's the sheer number, but also the diversity. I thought about how to demonstrate this diversity without getting way too technical and boring. So I thought I will do it first with something that we can all relate to, humans. And then I'm going to use that as a point of reference to demonstrate what we're dealing with in the gut. So the way living things are organized evolutionarily, it starts with a domain, and then it breaks up into kingdoms, and then into orders, and then families, and then genus, and then race. As you can see, it just gets more and more diverse. Now, let's take me. I'm Heide. I'm down here. And let's go backwards in this. Ethnicity, I'm Persian. Race, I'm white. Species, I'm sapiens. Genus, I'm Homo. Homo sapiens, I'm a human. I belong to the family Hominoid. I'm a primate. I'm a mammal. I have a backbone. I belong to the animal kingdom, and I'm a eukaryote, meaning that my cells have structures in them. Okay, so this is the backwards of this thing. Now let's go to gut microbiome. So I'm going to keep this as point of reference. This is what we're dealing with in the gut microbiome. We have fungi, we have bacteria, we have archaea, and we have viruses. Those are the different things that make up the gut microbiome. Of these, only fungi are eukaryotes. From even fungi, from there on, they diverge from us. The kingdom is even different. Bacteria and archaea are actually not even eukaryotes, they're prokaryotes. And viruses are not even considered living organisms. So you can imagine that if we wanted to take them from this step and try to work it down to an individual bug, it's going to be a tremendous job. We have made some headway. We have done the bacteria in PD, and we've been able to come down to the level of genus and species. And we are now trying to pinpoint it down all the way to the individual bugs. We actually have funding from US Army to do this to fill out this entire table, do the fungi, archaea. Not sure we'll have money to do the viruses, but we're gonna work this all down all the way to the bugs. So it's not only a matter of finding individual bugs, but also how bugs interact with each other, because they're not just out there on their own, they're in communities. So my lab has published 2 major papers on gut microbiome and PD. By the way, this is me down here. Just wanted to let you know that when you're last person on the list of co-authors, that means that you are the principal investigator. You're the big guy or big girl. So it comes out of my lab. That's what it says. And Erin Hill-Burns and Zach Wallen were the ones who did most of the work. Erin was a postdoc in my lab, and Zach was a graduate student who just defended his thesis. So in the first paper, we asked the global question of, is there a difference in the gut microbiome of persons with PD as opposed to people who don't have PD? And the answer was a resounding yes. And because people with PD take medications, and medications can affect the microbiome, we made sure that what we're seeing for Parkinson's was not a consequence of medications. So we showed that there are signatures that are distinct that go with Parkinson's and with the medication. Now, having established that the gut microbiome of Parkinson's is altered, then in our subsequent paper, 2020, 3 years later, we started to characterize what is it that has changed. I'm going to show you data right out of a paper. Bear with me. I'm not going to dumbify this. Oh, not yet. But this is actually data. So of all the factors, we looked at about 40 some factors to see what affects gut microbiome. Population studies, regardless of PD, have shown that gender and age and medications have significant effects on microbiome. So we detected those, but we also detected an effect for Parkinson's disease, which was actually bigger than gender and age. And believe it or not, geography has the biggest effect. This is really puzzling, but other people are showing that too. Okay, remember this one? Now, I'm going to show you the analysis that we did at this level. We have identified 500 at the genus level, 500 genera, 500 groups of bacteria in the guts of people who are in our study, be it they have Parkinson's or not. So I'm going to show you what this looks like, trying to characterize it. This is the paper if you want to go look it up. So each dot here is a genus of bacteria, a group of bacteria. The plot was made based on the correlation between different genera based on their relative abundances. In other words, it's the network. If they're connected to each other, if there's a line between them, they are in a cluster. They're making a network together. So this is just computer generated based on our data. And the colors show that we have one very distinct network up there, and we have three that are semi-connected to each other, but they are different somewhat. And we have these little guys here, okay? Now, when we analyzed the Parkinson's gut microbiome, we found that of all these 500 genera, 15 of them are altered in abundance in the gut of Parkinson's disease. 10 of them are reduced, 5 of them are elevated in PD. So to see what they are, we took this picture, made everything gray, and then put on it, superimposed on it, the 15 that are associated with PD. We have 3 clusters. The ones that are blue, this cluster has too high a frequency in patients with PD. So does this one. But this and that are not related to each other. And this is a cluster that's actually low in PD. Is that clear? I'm basically saying that we have identified 3 clusters of microorganisms that are distinct. One cluster is depleted. 2 of the clusters are elevated, but they're not related to each other. Good? Let me just look at people here and see if it's good. Okay, good. Thank you very much for shaking your head. Excellent, thank you. All right, so we move. Let's see who these guys are. Never mind the names, but there are 3 that fall in cluster 1. So the question is, what are these bugs? What do they do? The ones that are in cluster 1, they're opportunistic pathogens. What does that mean? That means that if they find their way into a sterile environment in the body, and if the person has a weak immune system, they can become infectious. So pathogens. The second group that is reduced in PD, they produce short-chain fatty acids. Those are good things. They're anti-inflammatory. They keep the inflammation down. They protect the lining of the gut. And they're not just reduced in PD. They're reduced in most diseases that involve inflammation. And they are somewhat a consequence of disease. The longer you have PD, the lower the levels of short-chain fatty acids. So the short-chain fatty acid producing bacteria are actually a very active area of research, because that might be amenable to treatment for a lot of inflammatory diseases if somehow we can boost their levels back up. Finally, we have cluster 3, Lactobacillus and Bifidobacterium. Guess what? They're sold as probiotics. Those are the main ingredients in the probiotics that you buy off the shelf. And they're elevated in PD. Why are they elevated in PD? We don't know. Are they bad? They could be bad. They could become opportunistic pathogens. But they could also be a response of the body trying to counteract what's going on in the microbiome. We don't know. So the short-chain fatty acids were interesting. The probiotics being up in PD has been shown by other groups as well. The finding of the pathogens was the most novel finding that we had in our study. And it's generated some interest. People are following it up. And we are wondering if they are the pathogens that Brock hypothesized that are actually the trigger of Parkinson in the gut, or maybe they just find their way in the gut because the lining of the gut is compromised. But you know what? They're not supposed to be in the gut in that high a number. So it's a new avenue research that we are rigorously pursuing. So I would like to summarize. Parkinson's is not one disease, and the chances are one bullet will not cure all of them. The fact that we're now accepting the fact that PD is not one disease and trying to look at it as brain down or gut up, this is actually good news, because we've been having these clinical trials that gets our hopes high. A lot of money gets spent, and just about all of them fail. By recognizing that Parkinson's is not a single disease and admitting it and start figuring out who has what kind of disease, maybe now we can actually separate people based on the type of disease that they have, and then do cleaner clinical trials and get better answers. Another reason that I'm optimistic is the fact that there are now the pseudo clinical trials that you can do online with the data that's available, and that can inform clinical trials before you start it. So one of the people who posed the question ahead asked me if I think that there's going to be a cure in the next 20 years. I would like to say yes, because we've finally started accepting that the traditional way we were looking at PD is not the way to do it. And we have people from different fields coming in and helping us solve it. So there is intensive research underway for therapies to stop PD. Gut microbiome is only one of them. It's a new and a complementary effort in this fight. It's not going to give us all the answers. The pathogens we've found in the gut, which Brock actually hypothesized first, may actually be the trigger of a form of disease that starts in the gut and moves to brain. I'm actually working with people who are working in animal models, and we're testing right here at UAB in the laboratory of Dr. Standard. We're testing to see if pathogens can actually cause PD in animal models. If that's the case, then we have a target that we can develop preventions on. We also know that imbalances in gut microbiome contribute to progression of PD, like the progressive loss of bacteria that make short-chain fatty acids. So that is a target for hopefully slowing or stopping the progression. So if we can understand the altered microbiome and figure out ways to fix it, we may be able to slow or stop progression of PD in some people. Now, there were questions that were posed to me ahead of the talk. What dietary changes would you recommend? I thought this question would come, and every time I give a talk, I check the literature again. The literature on diet and Parkinson's is all over the place. You have this paper that has thousands of people in it. It's so robust, it's so excellent, that tells you that a fiber-rich diet better than fatty diet. You get another paper, twice the sample size, twice the power, says, oh no, a fatty diet is better than fiber. One says keto diet, one says Mediterranean diet. So one, I cannot recommend one because I'm not a dietitian and I'm not a physician. Two, one should not recommend a diet even if we knew what would work for PD, because for each person, it depends on other comorbidities you might have. I mean, do you have intestinal problems? I mean, your doctor is the person to talk to. For me, if everything is equal, this is what I think personally. I think a heart-healthy diet is the way to go. A heart-healthy diet, there's no evidence whatsoever that has any adverse effect on Parkinson's, but we know that it's good for your heart and your brain, and it actually reduces your risk of Alzheimer's disease. So that's what I go with. Nobody asked me if they should take probiotic supplement, but people tell me that, you know, I'm taking probiotics, and I cringe on that. If you're taking probiotics, this is what I have to tell you. Please take the bottle to your doctor. And if you want, I can send you the paper that just came out. And every other study that was done in Parkinson's disease that shows that these so-called probiotics are elevated in PD. I don't know if taking probiotic is a good idea. It might actually be hurtful. Please be conscientious of that and talk to your doctor. Another question was how likely there will be a cure or a prevention that's in the next 20 years. Cure is a big word. An effective treatment, something that slows or stops the progression of the disease, I would like to say it's pretty likely for the next 20 years. Prevention is tougher. For prevention, you have to know when the disease starts. And if you remember the graphs that I showed you from the beginning, we don't know, it goes back like decades. Do we start at birth? Do we go in utero? If we can figure out the different subtypes of Parkinson's and then figure out which one is caused by what and at what stage of disease, then we can intervene and stop it there. Right now, we're looking at people who have REM sleep behavior disorder. That's a phase before PD, and trying to stop it there before it goes to PD. And that's a very active field of research right now. I want to add two things. One is H. pylori. H. pylori is a bacteria that causes ulcers. I didn't know I had H. pylori until I went for the first colonoscopy after I was at 50. And they said, oh boy, you got ulcers. I had no idea. And they put me on antibiotics for H. pylori and I'm cured of it. Now there's a very easy test to test for it. The reason I'm bringing it up is that having H. pylori, which is very common, 30 to 50% of people with Parkinson's disease have H. pylori and may not know it. H. pylori interferes with uptake of L-DOPA. So if you're on L-DOPA, go to your doctor and ask to be tested for H. pylori. And if you want literature on that you can take to your doctor, I'll be happy to send you papers. It's not our work, but there are a lot of papers that have shown that. Nobody asked today, but the question of fecal transplant comes up. So if the microbiome of PD has been altered and is not good, can we change it? There are actually fecal gavages that are being used for people who are infected with C. difficile, for example. I know in Turkey, they're using fecal transplant in the form of a pill that you would swallow after they use antibiotic regimens to get rid of what's in your gut and then repopulate it. FDA is looking into it. It's something that people are looking into it. The issues now are, actually the biggest issue is we don't know what is a good microbiome. A healthy microbiome has not been defined. That's one issue. And the second issue, whose microbiome will you put in yourself? This is just on the side note. I've been thinking about this, and I was thinking with the people in my lab, we don't go in anymore. We talk on the Zoom all the time. And we were just thinking about the idea of maybe we should start banking our microbiome every 5 years or so, our own. So if we get some sort of a disease, we have our own, and do a transplant. There you go. If we have any entrepreneurs in the audience, let's talk. Okay, that's all I had to say. So, and we have time left. If there are other questions, I'll be happy to entertain questions. But remember, I'm not a physician. I cannot tell you about the drugs you're taking, the doses, etc. I'm a scientist. Ooh, look at that. I'll be right back.
We go.
Boy, the sun is pretty.
You're back.
There we go. Yeah.
All right. Any other questions? I see you good now.
Yeah, I wish you had said something.
No, you're fine. You're fine.
Let's see, from Brian again.
Let's see. I think we have them. And there's one actually general question that definitely some context, a little more context probably be helpful. The question was, what is the MIBG? And
That
you wouldn't,
did you get that from the legend of?
Yeah.
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 It is a marker that you can detect the pathology in the heart. It's just a marker.
okay. Any other questions? Okay. Going once. All right. Well, thank you everybody for joining us. Our next
I have a
webinar will.
question.
Yeah,
Yes,
go ahead.
Jim. Yes.
Yeah.
This is Jim Rhodes. I had to have my colon removed in 2011 due to 35 or more years of uncontrolled ulcerative colitis, and I always had H. pylori and all kinds of issues. So my question is, are there? And I've been diagnosed with Parkinson's going on 5 years now. Are there studies going on that I could participate in, having been in that group, or is there something out there that I could be a part of?
Actually, the topic is very actively researched right now, and I can find out for you to see if there's anything in Alabama you can participate in.
Okay.
If not, I'll find you another one as close to us as possible. So, so you have had ulcerative colitis.
Yes, I had it for over 40 years. And in 2011, I had to have my colon removed. I have a permanent ileostomy. And I was diagnosed with Parkinson's in 2016, February.
Okay. If you could please send your information to Brian,
Okay.
your email address, I'll figure this out and email it to you.
Wonderful. Thank you so much.
Hey, Jim,
Absolutely.
you don't need to do that. I've got your info from Zoom and I can send it to Dr. Payami.
Okay, great. Doctor, I have a relationship with UAB. Dr. Standard is my movement disorder specialist. And I also have a relationship with Vanderbilt because that's where all my parts are at.
Okay. Your parts. Okay, so Vanderbilt
That's excellent.
and David Standard.
Yes.
Got it.
Well, that's where my
Standard
parts used to be anyway. So
Yeah, okay. Got it. David Standard is my close collaborator.
I'm sorry to hear that.
Why?
No, I'm teasing.
He's a really nice guy.
I know he is a really nice guy. And my wife, Carolyn, is a Davis Finney and a Parkinson's Foundation ambassador. We've met you a couple of times
Yes.
down in Birmingham. So thank you so much.
Oh, you're welcome. Anything else I can help with?
If you drink wine, being a fermented beverage, would that have something like a probiotic to it that would be harmful?
Okay. The probiotic, no, wine does not have lactobacillus or bifidobacteria. Whether wine is good or bad for PD, I can't tell you.
Okay, just curious.
I would go in moderation.
Oh, no.
Yeah. Yes. But then I didn't define what moderation is.
Oh,
right?
okay. Thank you.
Yeah,
That's right. That's nuanced and complex, just like
that's
Parkinson's.
right.
Well, I have been told that alcohol in the right amount has a positive, and I know this personally, has a positive effect on tremors. So, meaning it reduces the tremors.
You said that, not me. I don't have an MD. I can't speak to that.
No, I'm as a patient, as a
Yes.
party.
I've heard that. Yes, I have heard that. Yes.
I've tested it more than once. I'm Irish, so. Sorry.
No.
So if there are no more questions, I would like to thank you for joining me
Absolutely.
today.
Well,
This was
thank you
great
so
fun.
much.
If I can be of any help in the future, you know how to get a hold of me through Brian, right?
Absolutely. Parkinson's Association, and I will just let everybody know our next webinars. It will be, let's see, be after the first of the year, but it will be January 14th, and we'll have an announcement out on our websites and social media channels and newsletter. I would say probably sometime next week. If there's nothing else, thank you again for your time, Dr. Payami. That was great.
My pleasure.
Really,
Thank you,
really
Brian.
appreciate it. Oh, absolutely. We'll hopefully see you guys in the next webinar after the new year. Everybody have a great, safe holiday.
You too.
All right. Take care now.
Thank you.
You got it.